So, well, last time I have been speaking about apple scab, powdery mildew, fire blight, some nectaria, cankers, and crown and collar rot. This time I would like to speak about Alternaria mainly, then white ace and ramularia, which are occurring in the field and then causing issues in the post-harvest, brown rot, lenticel rot and bitter rot, Botrytis and penicillium. And then if we have more time, a bit about disease management strategy, or yes, we will anyway see about this. Okay, so the first I will start is Alternaria. Alternaria is a very widely diffused pathogen, normally associated with humid conditions. It's a saprophyte that you can find also on the walls of the house when there is high humidity inside. Anyway, it is also the agent of diseases, in particular, or of black spot and black rot. We see here some symptoms on leaves here and below on apple occurring directly in field. This is a disease that in the last years in our condition has been growing very much. We believe this is due to the climate change condition, because in the past it was not a very big issue. Now, year after year is growing in incidence and also in severity. And the conditions that are favoring Alternaria are mainly the increase of the average temperature, in particular during spring, and also the humidity conditions. So two factors that are favoring the growth of this disease. Here we see our isolations, in particular from leaf of golden orange apple, fruit of Sorino red, which is a variety resistant to apple scab. As you can see, sometimes the leaf spots or fruit spots are surrounded by also some redding of the margin. And when we inoculate these Alternaria on fruit, they reproduce the symptoms, but normally this is different. So it is a black rot that we find when we inoculate. So also pathogenicity tests are not very easy to be carried out. Okay, I speak also about another Alternaria, which is called Alternaria alternata apple pathotype. In this case, it's not reported until now in Europe. It is considered a quarantine pathogen. It causes spots on leaves and also on fruits, on susceptible cultivars. And in this case, the necrosis is accompanied by the formation of a toxin, a phytotoxin, which is called AM toxin. I think it's very diffused in Asia, also probably in China. Okay, and then we have another disease which is growing always in our area in Italy, northern Italy. We have black spot also occurring on pear. This was not in the past very present, whereas now it's becoming more and more relevant. Okay, so we did a monitoring. This is just in Italian, but to see how much this was diffused, and we could see that a lot of isolates that we found were virulent. Then to understand what is the species involved, we did some molecular identification of the isolates, and they mainly belong to two species or species complexes. One is Alternaria alternata, the other is Alternaria arborescens. Anyway, we must say that there are isolates which are not in one group or the other. So they are maybe new species, but identification and taxonomy inside Alternaria is very complicated. These are other symptoms on apple and pear. These are some isolates of Alternaria. And here you see under a microscope, identification is, at least of the genus, is quite easy because there are these conidia, which are dark, muriform, with a beak and multicellular. Very rarely in nature we can observe also the reproduction form, which is ascospores produced into asci and released by pseudothecia. There was a very good identification manual by Emory Simmons. It was written some years ago. Emory Simmons is not alive anymore. And here there were 276 species. Anyway, I must tell you that the taxonomy has been really changed in the last years. In the case of Alternaria, the inoculum source is plant residues into the field. Also, they could be fruit residues in post-harvest. And the conidia are dispersed by wind and rain. They can penetrate through wounds, mainly caused by insects, also by lenticel and stylar channel or calyx end. In this case, they cause core rots, as you can see from this picture above or below. And in this case, you can observe a mycelium inside, which is normally white, and it's really thin shaped. Okay, occupying initially the core of the fruit and then extending to occupy also the rest of the fruit. Well, this is taken from Agrios. It's very easy life cycle because it's mainly based on conidia. There are not reproduction structure, and normally first we have these lesions occurring on the leaves. Later in the season, we have fruits which are affected, also in the post-harvest phase. By considering the field control, until now, we still have several products available, but our strategy in the field is mainly targeting apple scab and then later powdery mildew. So we don't have normally specific treatments against Alternaria or Stemphylium. Stemphylium is a very close species to Alternaria, which is very important for pear production. Among the agronomical practice and equilibrated irrigation, some treatments with also biofungicides, and harvest in a dry season or a dry moment the fruit are good practices. In the post-harvest phase, it's important to sort the fruit, to select, to remove the rotten fruit, to have a careful handling, disinfect boxes, process line, and storage chambers, and have rapid cooling in controlled atmosphere or dynamic atmosphere. Last, which is also relevant, and the European Union is starting to consider this, is that Alternaria genus is also producing Alternaria toxins. Here we have a list of the mycotoxins which are considered by the European Food Safety Authority. So recently this year they released a new regulation for controlling and checking the level of these mycotoxins. The chemical structure, well, are very complicated, and a lot of metabolites are produced by Alternaria. They have effect mainly as immunosuppression and also infertility. They are also possibly involved in the development of esophageal cancer. And yes, we worked also on this by developing some HPLC method for detection of these Alternaria toxins in different matrices. Okay, so this is just to show Stemphylium vesicarium. It is the agent of brown rot of pear, and it's very similar to Alternaria, but for the symptoms. But if you observe under microscope, you can see that the conidia have a different shape. Well, they are still dark and pluricellular, but they do not have a beak as Alternaria. Okay, so now I move to a second group, in particular the agents of white haze. White haze is caused by some basidiomycetes, fungi. In this case, these fungi are producing ballistospores, which are released by sterigmata. It's a group of fungi which are very similar to yeast. So they are called yeast-like basidiomycetes. They belong to two orders, Enteromatales and Golubeviales, and they started to be reported since the last 10-15 years, first in the Netherlands, then in Italy, Germany, and Croatia. And what they cause is mainly a cosmetic defect. So it's not a real disease, but they create a waxy, opaque, superficial film on the cuticle of the fruit. And they are very slow growers. And, sorry, my computer is blocked. Okay. Okay, what we did is a long monitoring from during the apple growing season, from May to September, on the bark, leaves, fruits, and weeds of apple orchard to isolate these fungi. Then, together with an institute in the Netherlands, the Westerdick Institute, we identified the species. We have mainly Golubevia pallescens, Phylloxera washingtonensis, and Entyloma belangeri as agents of this white haze. We performed the monitoring in 3 regions of Italy. I know Italy compared to China is very small, but we have different regions where apple is cultivated. And what we found is that in every region there was a prevalent species. Okay, so with this white haze, we stopped without understanding very well how the fungus is arriving on the fruit. And that was our main question. But before answering the question, I would like to describe another disease. This is called dry lenticel rot. Dry lenticel rot is also another emerging disease which is growing year after year in Europe. It started in Italy, but now we have also reports in France and Austria. So it's a new pathogen which is occurring. The name of the pathogen is Ramularia mali, and it's affecting some susceptible varieties, for example, ambrosia, but also golden delicious, late during the storage. So this is a pathogen that is coming out from the lenticels of the fruit, but it shows a very dry rot. So it's different from other pathogens. It is also very difficult to be isolated. So in this case, again, the question was to understand where this pathogen is coming from. Is it coming from the field from the air which is present in the orchard, or it's coming from the fruit directly. So to answer these questions about the epidemiology of the pathogens, we tried to adopt a new approach which is based on metagenomics and microbiome study. So we took two varieties. One is the ambrosia, the other is the opal variety. And into this variety, we monitored the disease incidence throughout the season. You can see that at the end of shelf life, or at the end of storage, disease incidence was 20% for white haze and 15% for dry lenticel rot. So by studying at different time points the presence of these potential fungi, in particular Ramularia and Golubevia, so dry lenticel rot and white haze, what we found is that Ramularia was occurring as epiphyte, so on the surface, on the skin of the fruit, starting from August, then September, storage and shelf life. Looking at Golubevia, also Golubevia was occurring from September and then during storage. So it was on the susceptible variety. But our question was where is coming from, and the answer came from the study of the endophytes. So the endophytes are the fungi inside the apple, into the pulp of the apple. In this case, you can see that in June and July and also August, but little in August, Ramularia is really present as an endophyte inside the fruit. So it's really not a pathogen. Just in susceptible varieties and under favorable conditions, it becomes pathogenic by causing dry lenticel rot. And always from this study, you could see that Golubevia, the other pathogen agent of white haze, is never present. So Golubevia comes from the air in the orchard. And now we are trying to understand by spore trap where it is coming from. Well, I also add that we did from some microbiology study to understand the network of the pathogen inside the fruit. And just it's very interesting to show that, well, Golubevia has a very close correlation with the Ramularia. So the two pathogens are growing in very similar conditions. Okay, so this is about our study of the microbiome. Last, we developed a diagnostic tool for specific detection of Ramularia mali. This is Ramularia mali on Ambrosia and Golden Delicious. We isolate a lot of strains, and by doing molecular analysis, we found that it was only one species. Then we designed some primers. In particular, we targeted a very good region, Calmodulin region. And then, well, without spending more words, but we developed a real-time PCR approach for early detection of this pathogen. Okay, these are the results of Ambrosia and opal apples and the presence of this Ramularia mali on the fruit. Okay, so I now move to brown rot. Brown rot is a disease which is very present in stone fruit. So in Italy, we have brown rot on peach, apricot, plum, cherry. It's very important for this type of fruit, but much less relevant for apple. Anyway, I just show you that there are some varieties which are very susceptible, and in particular the rennet variety of apple is really affected. The pathogen in this case is developing already in field by rotting the fruit. It causes this brown rot, so it has different color from Alternaria rot, because Alternaria is more blackish. This is more brown, and under high humidity conditions, we can find these circular molds, which is caused by Monilinia fructigena. Monilinia fructigena is a species that has not reproduction form, differently from Monilinia fructicola. And just in 2021, we reported for the first time in Italy a new species, which is Monilinia polystroma. Monilinia polystroma is very similar to Monilinia fructigena, and it has already reported in China on plum, but not until now on apple. So it's also this is an emerging pathogen. Okay, so now I move to lenticel rot. As I told you before, there is a dry lenticel rot, which is caused by Ramularia. On the other case, we have here lenticel rot. Lenticel rot is more soft rot, always starting anyway from the lenticels. And it is caused by different species belonging to the genus Neofabrea. Neofabrea in the past was called Flictema and previously Gliosporium. So this genus has changed the name several times. There are different species, in particular Neofabrea malichicortis, Neofabrea alba, and Neofabrea perennens, and also Neofabrea kielholzi. So 4 species which are causing cankers, perennial cankers on the apple tree, but also lenticel rots, and this disease has some synonyms. So it is also called bullseye rot or Gliosporium rot. Here we can see some symptoms on pink lady apples. As you can see from the symptom, the middle of the rot normally is yellow, whereas the external part is brown. So for this reason, it is very similar to an eye, and so it has been called bull's eye rot, because it has this appearance very close to the eye of an animal. And when we look inside by cutting the apple, normally we can see a V-shaped rot, which is going towards the center of the fruit. Initially, the lenticel rot is very small, and then it enlarges, and the rots go to confluescence. So they fuse one to the other. Okay, this is a pathogen which is infecting the fruit already in the field, but it is a latent pathogen. Normally, it penetrates the lenticel or the wound. It can penetrate around the stem or the calyx, but it remains as as a latent pathogen for several months, and only during storage it shows the symptoms. Okay, so as we said, there are 4 main species reported in different countries. It's very reported in Europe and North America mainly, not too much in Asia. There are not reports until now in Asia. Identification in this case is quite difficult, but some genes can work very well for the identification. In particular, this, which is elongation factor alpha gene, and it's very suitable for identification. Then here we have just a table, which is in Italian, sorry, but we have the 4 species, the disease symptoms on the tree, these perennial cankers, and the disease symptoms on the fruit. In this case, we have bullseye rot or lenticel rot, depending on the species, but they are very similar. We have to say that there are some cultivars which are more susceptible compared to others. In particular, Crips pink. In Italian, we call it commercially pink lady variety. This is really susceptible. If you don't do some treatments at the end of the season, at the end of the storage, you can have over 70% of the fruit affected by this pathogen. Anyway, other varieties are also susceptible, so golden delicious apple and also Bosch pears. Here you see during the season from harvest through the storage, this was a study done in 2007, how the incidence is growing quickly during the season. Well, I told you that it is a quiescent infection, not occurring after harvest. The conidia are mucilaginous and born by the water, so after rain. Then the fungus normally stays on the surface, very close to the lenticels, and just when the fruits start to ripen, we can observe the symptoms. The fungus can also grow at -1 degrees. Normally, the symptoms occur after January storage. So we harvest in September. After 4 months, we have the first symptoms. Prevention can be done by pruning the cankers, pruning the plants, and then by cold storage and fungicide treatments. Fungicide treatments can be done with several products, but one of the most effective is Bellis, which is a mixture of boscalid, SDHI, together with piraclostrobin. These mixtures seem to work quite well. Okay, still about the biology, I add that it is a very difficult fungus to be studied because it is very slow grower. It's also difficult to have enough conidia when we grow it on artificial media. But we can see, we can start to know the first solution about this difficult pathogen because it is favored by the increase of pH. So when the fruit ripen and the pH increase, the fungus can release some alkalinizing compounds, and this favors the infection. In the orchard, Neofabrea can produce conidia in cankers and also in dead bark tissues. Then these conidia are dispersed by splash, so by water, and they can reach the fruit. Once they are in the fruit, they remain in a latent or quiescent stage for several months. In particular, susceptible varieties include Bosch, Anjou, and Bartlett Pear, and also Crips pink apple. In particular, fruit which are harvested very late in the season, for example in October, they are favored in the infection. Yes, a last aspect which is important is that the reduction of malic acid in the apple can favor the germination of the conidia. This is quite a recent result. Okay, I move now to another disease of apple. So you can see that in Italy we have a lot of diseases, and this is called bitter rot, or it is also called anthracnose. Anthracnose or bitter rot are caused by two main pathogens, which both are nowadays considered species complexes. One is Colletotricum acutatum, and the other is Colletotricum gleosporioides. I have to tell you that until a few years ago, bitter rot was only caused by Colletotricum gleosporioides. Now, since 10 years, we have a lot of reports of Colletotricum acutatum. It seems that acutatum is taking the role that previously was belonging to Colletotricum gleosporioides. Okay, so acutatum was reported for the first time on apple in 1997. At the beginning in Europe, it was considered a quarantine pathogen, and then it was removed a few years ago, in 2008, because it started to be widespread everywhere. We also have an hypothesis that Colletotricum acutatum could have moved from strawberries, because initially it was only pathogen reported on strawberry, and then it moved to several other crops. Where there is Colletotricum acutatum, it's important to do spray treatments at 10 or 14 days interval. I should say that in Italy, it's not the target pathogen, because as I told you, we mainly have Venturium equalis and powdery mildew. But in other parts of the world, for example, in New Zealand, it's very important to do these fungicide spraying programs. Okay. Yes. Sorry, I moved in the wrong direction. Okay, here we see another symptom on fruit. The disease occurring on leaves and fruit is called glomerella leaf spot. It has recently been reported in France, but also in Italy. You see here Colletotricum. There are also other species of Colletotricum. So in the past, we had only Gleosporioides, then acutatum. Now we have also Colletotricum fructicola, first reported in 2019, and also Colletotricum godetia, first reported in 2014 in UK. In our region, in northern Italy, we have Colletotricum fiorinea, godetia, and acutatum on different apple varieties. I show you some symptoms from the fruit. First, we have some small lesions, which are dark, sunken. Then we move to have some larger lesions. Sometimes the spots are surrounded by red color. So this is very similar also to Alternaria. When they become enough larger, they produce the multiplication form of the acervuli. And the acervuli normally can be very easily seen because they produce conidial mass, which are orange in the color. If the pathogen is also occurring in the distal end of the fruit, it causes what is called eye rot. This is about Coleotricum acutatum. Coleotricum gliosporioides is a bit similar, but the conidia and the peritaceae, which are produced, normally form a cream salmon conidia. So the big difference is also in the color. In one case, it's orange for acutatum. In the other case, it's light cream for Coleotricum gliosporioides. Okay, so the fruit which are severely affected can drop off the fruit, or they can remain in the plant canopy as mummified fruit. Yes, I also show what is happening in post-harvest. In post-harvest, we can have bitter rot symptoms. Here we have Royal Gala apples, and they are affected by Coleotricum acutatum at the end of storage. You can see that lesions are similar, circular. When they grow, they converge, and at the end they overlap. And again, we can see the acervuli producing these orange conidia in ring patterns. Okay, this is other fruits inoculated. In particular, we can observe in this case the V-shaped form of the wound inoculated apples, in this case Royal Gala. So, about these two species complexes, Coleotricum gliosporioides and Coleotricum acutatum, the taxonomical approach used is based on multi-locus sequence typing, sequence analysis. The fungus, the two species complexes are divided into phylospecies, but currently there is not any biological mean for these phylospecies. So it's better to consider the two species complexes, acutatum and gliosporioides. Acutatum can be differentiated by gliosporioides because it has a slower growth rate on PDA, Petri dish. It has a fusiform conidia compared to the cylindrical conidia of gliosporioides. It produces orange colonies, whereas gliosporioides produce white creamy colonies. It's also acutatum less tolerant to high temperature. It is tolerant to benzimidazole, such as benomil, carbendazim, or thiabendazole. And the sexual stage is present for Coleotricum gliosporioides, but not for Coleotricum acutatum, or at least is very rare. Here we have the disease cycle, which is normally starting in the spring from cankers in the canopy of the plant. Here we have ascospore or conidia released by the rain, and these go and infect the twigs or the branches of the plants by forming cankers. From the cankers, later in the season, there will be conidia release, which are then infecting the apple fruits. In particular, rain is favoring this infection, and the conidia normally reach the surface of the fruit. They remain latent, they form an appressorium, and just later they can germinate. Okay, so here we can see the cankers which are forming, also maybe from the rotten fruit. They differentiate the peritaceae, the reproduction form. Ascospores can infect the fruit or leaf, or again twigs. And then there is a first penetration with the latent infection, formation of the appressorium, and then later invasion of the fruit and formation of the acerbuli. Okay, the disease cycle of Coledotrium acutatum is quite similar, but in this case, the inoculum, this is a study mainly performed until now in New Zealand by the group of Kerry Everett. So in this case, inoculum is released by the buds which are opening during spring. There is an infection of the canopy, and the bud scales or flower petals will go to the soil, to the ground. Then later here there is the development of the conidia. The conidia by rain splash, so during rain, they will infect the buds and the leaves. And this occurs when the temperature is around 15 degrees with at least 3 days of wetness. Okay, and this is what is reported by this paper, where, but you have to consider New Zealand is in the other part of the globe, so in the austral hemisphere. So in this case, we have a winter in the buds or cankers. In spring, we have first petal or leaf buds which are infected. They go to the soil. During summer, there is infection of the fruit, and in March, we can observe the development of this bitter rot. Okay, I move to the last group of pathogens. We have now Botrytis, and Penicillium. Well, about Botrytis, I have to tell that it is considered nowadays more a model pathogen. So it's not so relevant for apple fruit. Anyway, we still have some attacks. Compared to the past, we have a much less Botrytis development. It is the agent of gray mould rot. The species is called Botrytis cinerea. Cinerea means gray. And the incidence normally at the end of storage is 1–2% of the fruit affected. Anyway, on susceptible varieties, we can have also higher incidence. The infection starts from germination of the conidia, which can have some primary lesions, and then we can have also some secondary lesions. So in this case, normally the disease cycle starts with sclerotia. Sclerotia are really relevant for the overwintering mycelium. Then the sclerotia are germinating. They produce conidia, and the conidia can infect flowers and petals of the flower by creating a necrotic infection with collapse of the tissues, and finally we have the blossom and the rot of the apple or the gray mold. Sometimes it's possible in laboratory condition to have the formation of the apothecia, so the reproduction form from the sclerotia, but more often, normally in nature, we observe the conidiophores. The conidiophores, which are producing conidia, and the conidia can infect. Conidia are transparent and unicellular. Here is the cultivation in petri dishes, where you observe the gray mycelium and the sclerotia, which differentiate in the middle. Normally, this pathogen is penetrating through the wounds, also through the peduncle on pears, or also by nesting. So from one diseased fruit to one healthy fruit. And on apple, the main inoculum source are the plant residues, where the fungus can survive as sclerotia. Also in post-harvest, the main infection source is the presence of rotten fruits, in particular on the boxes or processing lines or cold storage rooms. So cleaning and disinfection are very important aspects. The conidia are airborne, so just some movement of the air can transport them to quite long distances. And the fungus can grow at best temperature of 20 degrees, but it can also survive at -1. So even in storage of apples with very low temperatures, we still have problems with Botrytis cinerea. In a field, Botrytis can infect twigs by causing cankers, or young fruits by causing a dry rot, in this case circular and surrounded by some red halo. After harvesting, we can have infection by contact, and there can also be some rots at the calyx end, which cause skin reddening, and later also some soft rots inside the fruit. So for Botrytis cinerea, prevention is mainly elimination of infection sources in fields, so twig cankers, use of anti-botrytis products after flowering, careful handling of the fruit not to cause bruises or wounds, disinfection of the environments, controlled atmosphere, because carbon dioxide has an inhibitory effect on Botrytis cinerea, and also treatments with benzimidazoles in post-harvest, in particular thiabendazole. Okay, I now move to blue mold. Blue mold is the last pathogen of this long overview. Blue mold of apple and pear is caused by Penicillium expansum mainly, but also by other secondary species of Penicillium. For example, Penicillium solitum, commune, verrucosum, grisefulvum, chrysogenum. So all these have been also reported. Penicillium expansum causes blue mold, so direct problems, but also it is the producer of pathoulin. And pathoulin is a mycotoxin which is very dangerous and regulated by the European Union. So fruit juices, apple juices, which are sold in Europe, cannot overpass a certain limit. So Penicillium expansum is a very big issue also for this mycotoxin production. On apple and pear varieties, which are more susceptible, up to 30–40% of the fruits can be affected. In particular, the most susceptible varieties are Conference and Kaiser, in the case of pears, and Ambrosia and Golden Delicious in the case of apple. Also, this fungal can grow until -3 degrees, so very low temperature, even below 0. And yes, Penicillium, well, it is a terverticillate species. This is how it appears on Petri dish. The symptoms, initially, we can see some small brown to yellow spots, which are circular, humid, and with very well defined edges. Then the rot grows very quickly, and it can occupy the whole surface of the fruit. It causes a very soft rot, From the lenticels, we can observe the white mycelium, and sometimes in high humidity conditions, we have also the occurrence of the blue-green conidia and conidiophores that give this color. It's a pathogen necrotrophic, which is growing very quickly by causing a deliquescent rot that it is very circular. Normally, the blue mold rot is very clearly delimited by the healthy part of the fruit. But studies have shown that pathogen can also present in the other part of the fruit. So it can be present also on the healthy part. The pathogen is a wound pathogen. It penetrates mainly through the wounds, but also through lenticels, stem, stylar channels, and in this case, it can cause again what is called the core rot. It is favored by previous pathogens or infections in the field, so apple scab, powdery mildew, also some insects can favor penicillium. In the post-harvest phase, mainly the sources of inoculum are the residues of rotten fruits in packing houses, processing lines, and cold chambers. In this case, there is a strong role by the pH, but this fungus is an acidifier fungus. So it's the opposite of Neofabrea. Neofabrea was alkalinizing the medium. In this case, penicillium is increasing the acidification and lowering the pH. So you can see decayed fruit always have a pH lower than healthy fruits. Why this pH reduction? Mainly by the production of gluconic acid. Gluconic acid is produced starting from glucose through the action of glucose oxidase. So we have this pH reduction. During acidification, the fungus produces some effectors. In particular, there are some Lys-M effectors, and these Lys-M effectors are able to bind the chitin of the fungus to protect it against the chitinases which are released by the plant. So it's a mechanism of protection against the plant resistance. Okay, and finally, there is the production of pathogen. Pathogen, which is a very small organic molecule produced by Penicillium expansum and other species. It is highly toxic. In particular, it causes acute toxicity, so agitation, convulsions, intestinal inflammation and vomiting, and also chronic effects such as genotoxicity, immunotoxicity, and neurotoxicity. So the European Union established already in 2006 sorry, some limits, and the limits are 50 parts per billion in fruit juices, whereas in children or infant, the limit is 10 parts per billion. So, to reduce pathogen, we should avoid to harvest the fruit in a wet and rainy period. It is important to keep the cold chain for the fruit and to eliminate wounded or attacked fruits. So prevention is based on careful handling, careful sorting, hygienic practices, and then storage and packing house practices with low temperature and controlled atmosphere. It's also important to do some phytosanitary control throughout the season. And if necessary, it's possible to apply also some chemical control, some physical tools of storage, or also biocontrol agents. So now I believe I am more or less at the end of my time. I just leave one slide of the last part, which is about post-harvest disease management, by saying that it's very important prevention, mainly, also disease monitoring, so having some good tool to identify the pathogen and disease occurrence, and disease control. Disease control in Europe, it's moving in the last years towards the elimination of the chemical fungicides. So we are really in a complicated environment because we try to find some alternatives. And so hot water treatments, natural compounds, biocontrol agents, and resistance inducers are the strategies that we are trying to develop and to put on the market in order to find some good control practices.